Can everybody hear me online? Wonderful. And welcome to our Zoom friends. My name is Edward Nagel, and I'm the co-president of Technion Canada. Thank you for being here with us today. We are gathered in several homes, including in Toronto and Montreal, and we're excited that many of you are joining us by Zoom to learn about this amazing alliance. So let's get started on the main event. We will start the Zoom link with a brief video and then hear directly from our Technion leadership in Israel and our fantastic researchers. Despite the extreme challenges happening in Haifa, we are so grateful that you have taken the time to join us for this event. Please remember, for those on Zoom, to mute yourselves throughout the presentation and to write any questions you may have in the chat. Those of you who are live, please note your questions, as there will be time in the Q&A for each host home to submit its questions. Please enjoy this brief video, following which I will turn the floor over to Professor Wayne Kaplan, Vice President for External Relations and Resource Development of the Technion. And now, the video.
Have you ever wondered what the world would be like if Technion didn't exist? Imagine the state of Israel maturing without technological acumen. Perhaps the most urgent requirement of any free country wishing to preserve its standing, dignity, and independence. Without the generations of students, researchers, lecturers, and graduates instilling a fundamental approach to science, from which groundbreaking engineering leads to a cutting-edge microelectronics industry, leading architecture and town planning, pioneering building and transportation infrastructure, revolutionary security and HLS industries with secret developments beyond our imagination, an entrepreneurial spirit that is second nature to the trailblazing startup nation. We are fortunate to have Technion Israel Institute of Technology. Join us in celebrating the Technion Centennial Year and the next fascinating 100 years.
I think you're up.
Okay. That's very clear, but okay, I think. Well, good evening or good afternoon, everyone. Thank you for making time to join us by Zoom in this way. My name is Wayne Kaplan. I'm a professor at the Technion in the Department of Materials Science and Engineering. So I'm probably the only non-life science person attending from the Technion at the moment. But I hope they'll allow me to be here anyway. I also serve as the Vice President for External Relations and Resource Development. So it's a wonderful opportunity to meet with all of you today and tell you about this exciting development that's going on campus. I should mention that it's tough times in Israel. I think everybody knows that. Today we had the formal national ceremonies honoring what happened on the date of October 7th last year, also at the Technion. And as everybody knows, this is still going on in Gaza and in Lebanon. And unfortunately, we had bad news today. Another Technion student was killed yesterday and also last week on Thursday. So these are trying times for all of us here. On one hand, and you may ask, this is surreal. I mean, you have a war, you have, how do you function? There's missiles coming in. And by the way, we may be interrupted. That happens, as people know here. And it is a little bit surreal on one hand. But that's Israel. And that's not new to us. And it's not new to us at the Technion. And I should say that the Technion has a very real role in dealing with the issues today. We provide means to to help the country in a number of different ways. And most of them are focused on what we'll need in the future. And we all know that when this ends, and it will end, we pray as soon as possible, but it will end, then it's the Technion's responsibility to help kickstart the economy of this country again. And we've done that before, and we will do it again. So we have a number of projects that are based in this fundamental science of the Technion, but looking forward into implementation. And this evening, sorry, this afternoon for all of you, this is exactly what we're going to talk about, is a unique meld of fundamental science to applications, and in this case, in the medical field. So I'm particularly excited about the Wolf Center and what we're about to hear about. I think it has enormous potential impact. And we know that we can't do it without support and engagement with people around the world. And so it's a good opportunity to already thank those of you in Canada who have been involved in this project and in other projects on campus. I'm going to hand this over to Professor Noam Ziv and introduce him. Noam was born in Vancouver, heads the new Technion Human Health Initiative, and as a result, is the Deputy Senior Vice President of Biomedical Science and Engineering. He's also a member of the Ruth and Bruce Rappaport Faculty of Medicine. He's also a member of our Network Biology Research Labs, which is a cluster of 7 groups of researchers from 4 Technion academic departments. His diverse interests make him the perfect choice to spearhead THHI, the Technion Human Health Initiative, which conducts human health research in a comprehensive, integrative, and institution-wide manner. The new initiative brings together Technion scientists from multiple academic departments with researchers and clinicians from Technion-affiliated hospitals to meet some of the biggest challenges that we as human beings have. THHI will also partner with industry to convert the most promising research into applications and products that will serve the medical system and medical teams on the front lines. This is what we do at the Technion, is a rapid conversion of fundamental science to real applications. And I am convinced that Noam will spearhead this in this field as well. The goal is, of course, to build a bridge between medicine and life sciences, exact sciences, engineering, data science, and design. In this way, 1 + 1 is equal to 17. It's a high impact project, which Noam is. So I'm going to turn this over to Noam at this point. Noam, cheers.
Thank you. Thank you, Wayne. So I have with me here Professor Amit Meller, who's from the Technion Faculty of Biomedical Engineering, Professor Lior Gepstein, the Director of Rambam Healthcare Center, Division of Research and Innovation, and the Director of Cardiology, and Danny Eitan, Professor Danny Eitan, the Director of the Rambam Pediatric Critical Care Unit. All 4 of us are members of the Wolf Center Steering Committee, and we wish to share with you our vision for this new and unique center, which was made possible through the generosity of Larry and Andy Wolf. So please, I'll just share the presentation here and connect. I hope you can see this. Can you see this? Okay, good, perfect. So, well, what is the Wolf Center? Rambam is the largest healthcare center in the north of Israel, with a remarkable history of innovation and entrepreneurship, whereas the Technion is a leading academic research with, as you've seen, a remarkable history as well. So with the establishment of the Technion Human Health Initiative, it was only natural for these 2 institutions to come together. Our conviction is that Rambam, with its really acute understanding of clinical needs, many ideas that flow out of it, and being a wonderful test bed for clinical trials, Together, the Technion, with its history of discoveries and technologies and the ideas, together will give rise to solutions. So our vision for this institute, which will be sitting in these 2 floors in the New Hensley Health Discovery Tower, which is in the Rambam Healthcare Campus, is that it will serve as a vibrant research hotspot for nurturing ideas in the realm of human health and fast-tracking them to real-world solutions. In essence, we view this as workspaces containing experts and resources within which clinicians, scientists, and engineers work together to identify needs, propose solutions, develop prototypes, and test them in clinical settings. The audience for this place are entrepreneurially minded physicians, healthcare professionals, scientists, engineers, and students from the Technion, Rambam, and local high-tech hubs. We envision the center of being comprised of 6 workspaces or laboratories, as we call them. AI in medicine, which you'll hear about from Dani Eitan. Tissue engineering and regenerative medicine, which Leo will talk about. Biochips, which Amit will talk about. In addition, 3 more labs: medical device innovation, drug delivery and molecular therapy, and healthcare operations, which we will not talk about today, but feel free to ask questions about them later. In addition, the place should contain meeting rooms for interactions. We have space for prototyping a small series production company, offices for guests, examination, biosample facilities, offices for experts, etc. So we've been working for the last few months on laying out the floor plans. And what you see here are the actual floor plans for this place. You might notice there's no real barriers between the various labs. It's like one continuum. It's divided into spaces which fit what we talked about. We have the examination and biosample rooms, a drug delivery and molecular therapy lab, lots of places for interactions, the healthcare operations lab, and AI and medicine lab. The other floor, the floor 9, also has 3 laboratories on it. We have the medical device innovation lab, the tissue engineering and regenerative medicine lab that Leo will talk about, and the biochips lab, which Amit will be talking about. Now, interestingly enough, you'll see that there's an empty space here, and there's a reason for that. We are hoping to integrate a deep tech company that will basically help us to transform ideas into real-life products. We were this close to having perhaps the leading European company setting up shop in our center, and then the war came and kind of ruined our plans, but we haven't given up hope, so hopefully this will happen in the future. So just to give you a feel of what it looks like, here's the tower. It's actually standing, it's already there. And 2 of these floors are going to be where the center are. So this gives you kind of an architectural view of what it looks like. This is the drug delivery and molecular therapy lab, looking from both sides. We can't show you all the fancy machinery that's on the left because it is just an architectural rendition. This is the entrance. And then we move in, you'll see there's lots of spaces for interactions between physicians and engineers and students and scientists. These are healthcare operation labs, which is basically a dry lab. And on the other side, we find the AI and medicine lab, which Dani will be telling you as soon as this animation is over. And Dani, I'm handing this over to you. I'm going to give you control of the screen. I manage.
Hi. Just let me know if you can hear me well.
Dani, you have control.
Okay, can you hear me well?
We hear you, Danny.
Yeah, we can hear you.
Okay, I have no idea how I can control this.
Just click on the screen.
Hang on.
Yeah. Oh, okay, so it actually works. Okay, perfect. So thank you for listening. My name is Dani Eitan. I'm here wearing 2 hats, a clinician and a scientist. As a clinician, I'm a pediatric critical care physician. I head the pediatric critical care unit in Rambam, which is a very hectic 15-bed unit, which is a mixed unit. We take care of babies and infants all the way to young adults well, I'll talk about it a little bit later. And I also am, as a researcher, I'm a clinician scientist. So as a researcher, I'm part of the network biology laboratories with Noam Ziv, studying mainly applications and translational physiology to try to translate insights from the clinical side and basic science together to actually build tools to help our patients. And I'm part of the Technion-Rambam initiative for AI in medicine. So when we talk about medical AI, artificial intelligence, machine learning, big data, whatever you choose your buzzword, there's a lot of hype. There's a lot of academic activities. There's more than 150,000 papers published so far on the field from the last, I guess, 20 years. But when we go to the bedside, we don't see AI in medicine really transforming the way we take care of our patients. And there are many reasons and many gaps that actually prohibit us or are barriers to deploying AI tools in the medical field. Some of them are related to, yeah, I'm trying to get this going on. Oh, so yeah, there's just a delay. Okay, so some of them are related to an inherent gap between clinicians and the actual researchers and the scientists. Either physical gaps, they don't talk to each other, they don't even know, they don't have interactions. And some of them are related to the fact that we kind of speak different languages, clinicians and scientists. And it's sometimes, even if you get us in the same room, because we don't have the same background and domain knowledge, it's not that easy to get collaborations going, even if we do have the will to do it. Another kind of gap or field of gap is related to access to well-curated medical data. So medical data is usually hidden in silos because of regulations, different types of barriers, and researchers, in order to develop tools, need access to medical data, and not just medical data. They need medical data that it's clean, because otherwise you get what we call garbage in, garbage out. It has to be well annotated, and such medical data is hard to come by for many researchers. And last kind of theme of gaps is related to the fact that even if we do get researchers and clinicians working together, and they do have access to data, and they develop a tool that seems a priori really good and full of potentials, to actually implement and evaluate a tool in a prospective manner is really hard. There's a lot of barriers actually preventing us from taking a tool that's been published and trying it at the bedside to see if it can really help patients. And these three types of gaps have traditionally been very prohibitive to getting AI-based medical decision tools to the bedside. And if we really want to make a difference, then it's not just about collaborating about the science, it's about closing the loop from an idea all the way to the bedside, deploying those tools in a prospective manner under clinical tests and studies, and seeing, can we really affect the patients? So kind of lucky for us, in Technion and Rambam, we're kind of in the perfect storm in some sense. The Technion in itself is an AI leader, medicine and any other fields. It's actually been ranked the first in Europe in terms of AI research and productivity in several rankings. That's on the Technion side, and there's a lot of researchers there that are interested not just in doing AI, but doing an AI in medicine and impacting care. And on the other hand, Rambam is kind of the perfect partner in the sense that we have our own EMR, we have a large biobank, and we're poised to collaborate. And we've built a consortium, Rambam and Technion, trying exactly to capitalize on our mutual or individual advantages to see together how we can do this and start to build tools for AI in medicine. I'll jump because I've been told that we need to be quick. We have formed Terra, which is Technion-Rambam initiative. We've started with several joint products. We've allocated seed funds to several projects. I'll talk about it. We have a small research space that's way too small for us. We have several undergraduate projects going together, and we even have a course for healthcare professionals and for Technion students, 2 different courses related to AI in medicine. Several of the projects are related to ICU, to cardiovascular care, to building clinical decision support tools, to even helping patients in rehabilitations, which sadly the last year has given us a lot of patients to work with, things related to nutrition, oncology, all over the place. These projects are already ongoing at different stages of development. And I've been asked to talk a little bit about our own or my own personal bias or personal research. So I come from critical care, and specifically from pediatric intensive care. We take care of the sickest babies and children that for some reason have critical and complex illnesses. Very often they have more than one physiological system that's sick and disturbed. And these patients, they're very unstable. Their state can fluctuate over seconds to minutes. Every patient is surrounded by an array of monitors. We're a heavily technology-dependent field in medicine. You can see examples here in the pictures. Sometimes you can't even see the patient from the myriad of devices that engulf them. Some of these devices are related to monitoring. Other devices are related to actually stabilizing the patients and providing care, from infusion pumps to dialysis machines to an ECMO machine, which a couple of patients here in the pictures are on, ventilators and so forth and so forth. Not only every patient is super complex and dynamical, we have several patients we have to take care of the same time, up to 15 example for our unit. Other units in the world deal with up to 40 patients, with hundreds of staff involved in the care of a unit at any given time point. And so we have these patients that are, we say, heavily monitored, sampled from multiple dimensions, and receive a lot of care, again, for different interventions. And this is kind of the ideal or the dream playground for an AI application or for AI-based tools, because this is exactly where they shine. A lot of data at different frequencies, different modalities, and there's a lot of potentials for AI tools to help clinicians, not to replace clinicians, but to help us take care of the patients in a better way. And what my group, together with multiple partners at the Technion and also internationally, have been doing is trying to build patient-specific models that are based on AI and other dynamical fields together with, so these are patient-specific models that talk with all these monitors and devices, together with what we know about the population of similar patients and the biomedical knowledge. And these models are designed to help us better monitor our patients and try to choose the right therapy for them in an AI-aided manner. Thank you.
I will now pass the baton to Amit Miller, who will tell us about biochips. Amit, I'm going to give you the controls. Here we go. Okay, you have the screen.
Okay, great. Thank you, Noam, and good afternoon, everyone. It's a pleasure to meet you all. I'm coming from a physics background, and in fact, I became a biophysicist about 30 years ago, switching to biomedical engineering later on, and spent many years in the Boston area, where I had a chance to start working with MDs and doctors. Coming back to Israel around 2010, And I want to tell you a little bit about the biochip. So first, what are biochips? If you can think about biochip as a physical device that can actually replace anything that we do on the bench, on the wet bench, whether to prepare a biochemical reaction, measure biological molecules, diagnose a disease using a molecule. Anything that you can do on the bench, you can do on a chip. And the advantage is that it requires much less material. It can be transformed to the bedside. You just saw Danny's ER room filled with instruments. And our vision is that someday everything will be replaced with chips, which are small, compact, they require much less materials and have the ability to integrate much more sophisticated sensors on them. So in our unit, in the Wolf Center, we will going to target many different types of biochips. For example, wearables and in-body chips. Those are chips that you can wear on your wrist or somewhere else and monitor your certain measurements in your body for disease. We will develop very sophisticated sensors. We will do organ on chips, which is another frontier in this area. And the idea is that there will be a lab that will be able to support very vast different types of materials development, different fabrication types, and a set of experts that would be able to support any innovation or any initiative between the Rambam and the Technion. And we have a lot of ideas that requires the expertise if they can fit on a biochip. So to give you a little bit example for this versatile environment, so this environment in the Wolf Center will be able to help in design and fabrication involving both micro and nanofluidics, biochip integrated circuits in electronics, data management, using low power simulation for chips. We use many different tools these days to simulate things before we even start fabricating. The interface with biomaterials, anything that is needed to create those chips from the ground up and do it very quickly will be there to support the researchers. Just to give you a little bit idea of how it looks like, I prepared a few slides from my own lab telling you about some of the sensors that we develop for biochips. This sensor particularly is called nanopore, is a very, very small, a few nanometer size pore that is about the size of a single DNA molecule. The DNA molecule is about 2 nanometers apart. It's a very, very tiny molecule in a very long polymer. And what we can do, we can use electrical field in order to take these long molecules and thread them very linearly as a single file through those nanopores. And while they're threaded, we can read detailed electrical and optical information from this DNA molecule. We can integrate those devices in a biochip. This is a chip that actually takes a full human gene, million of base pairs of DNA, which every one of our cell contains, and stretch those molecules through a pillar array. You can see here in a basically real-time movie of a single DNA molecule going through this array, being stretched and then threaded through those nanopores that you can see here for further analysis. So these days, we only need one molecule, one piece of DNA in order to learn about problems, in order to learn and diagnose disease. And this is something that we are doing. Another type of chip, another area is in proteomics. Maybe you heard about it. This is one of the rising fields and will be a revolutionary area in the human proteome that we see coming in the next years. And here you see a chip that we've made that allows single protein sensing in in a tiny chip. You can see here thousands of proteins. Each dot here is a single protein. We can then follow those proteins and almost like give each protein a name here. You can see a tag. Each protein is now being tagged. And from the dynamics of those proteins and a few colors that we are sensing, we can identify those proteins. These are called biomarkers. They report on disease, and we can, in a few minutes, give a result, for example, associated with cancer or other neurodegenerative diseases that we are working on. So this is just to give you a little bit of flavor. What are biochips? What are these sensors? And what we think is the future of this whole area? Now, going back to our biochips lab in the Wolf Center, the main idea, and Noam touched on it, is basically to fast-track innovation, is to allow all our researchers, MDs and researchers, which come with a great idea to answer a specific need in biomedicine, to design the optimal chip, and with the experts in the center, make the prototype in the center, have access to all the computing resources, including AI, and perform preclinical trials, and basically bring it to a point where now you can interface with a startup. You can basically take it on to continue the chain. And we see the whole labs, and in fact, this network of labs in the Wolf Center as a one unit that basically gives, it's a one-stop shop, basically, that gives the answer for these innovations. And with that, I will end, giving it back to you, Noam, and be happy to answer questions.
Thank you very much, Amit. Our next speaker is, our last speaker also, is Lior Gebstein. I will pass the controls to you, Lior, and take it from there. Just 1 minute.
Okay, thank you. Thank you very much. I hope you can hear me. So Danny had told you that he has 2 hats. I'm a cardiologist. We always want to be better than the intensivist. So I have 3 hats. One is that I have a big stem cell lab at the Technion Faculty of Medicine. The second is that I'm director of cardiology at Rambam. And the third, that I'm in charge also on the research innovation in Rambam. What I'll try to do today is to tell you about the tissue engineering and regenerative medicine lab within the Wolf Center that we aim to develop. How do I control it? I'm clicking and nothing happens. There. So the aim of our center is to provide workspace and for experts and resources to allow collaborations between the physician who come up with the problems, the unmet needs, scientists and engineers, and industry partners in order to try to fix organs and tissues by developing new treatments within the field of regenerative and precision medicine using techniques such as stem cell tissue engineering and molecular biology tools. And the tools that we use are first pluripotent stem cells. These are stem cells that can come any cell type in the body. And we are using a specific type of pluripotent stem cells called iPS cells, in which you can create them from the patient itself. We're using a technique that Amit touched a little bit about it, is organ in a chip. The idea is to use the different cell types, liver cells, heart cells, lung cells, and so on, that we are developing from the stem cells to develop a new approach to develop drugs, to test drugs, in an aim to replace animal studies. So in order to develop a drug, instead of testing it in animals that are not similar to human tissues, to actually do it on human samples, the appropriate human samples. And the third technology is what we call tissue engineering and bioprinting. And the idea is to take the cells that you generate from the stem cells and together with unique bioinks, actually try to print tissues and organs to try to repair damaged organs. And I'll give you examples of the few things that we can do in our center. For example, we can, as I told you, make different cell types. Here are examples of heart cells that we make. and bronchial epithelium cells that we make in order to try to simulate the airways of the lung. This is an example how we use bioprinting. This is a picture from my lab in which we are taking the heart cells and trying to bioprint parts of the heart. And this is an example of an organ on a chip in which a alveoli, the terminal portion of the lung, is developed in order to test different drugs and how they affect the lungs, how they can diffuse through the lungs, and so on. So in order to tell you how this is supposed to work, I'll tell you on my own research, on my own field, but the same approach that I take for the heart can be for the liver, the brain, the pancreas, and so on. So first, we have to identify the problem. And the biggest problem in cardiology today is heart failure. We know that the heart cannot regenerate itself. So when you have an injury like a heart attack, this is irreversible. The dying muscle is replaced by scar tissue, and the heart as a pump doesn't function as well. And heart failure is a huge problem. For example, it's responsible for more hospitalizations than all forms of cancer combined. In its most severe form, the only solution is to replace the heart with a new heart with organ transplantation, but of course, we don't have enough hearts. So the idea is maybe to try to replace the heart with new heart cells. Now, can you try to move forward? It doesn't move. Okay. So the question, where we'll get the heart cells from? And here about 20 years ago, you could see Professor Itzkovich, who was the head of our OBG department at Rambam and also a Technion scientist. So together with the University of Wisconsin, they were able to create the first human embryonic stem cells for in vitro fertilization embryos, in which cells that can be now grown indefinitely in the lab have the capacity to generate any cell type of the body. And again, I don't have control. Can you click on it, Noam? So in my lab, we had early access to these cells. And as you can see, about 20 years ago, we were able to get the first beating human heart cell. It doesn't always project well over there, but you can see the beating area of the first human heart cells that were created. Next. We then showed in animal models that if we transplant these heart cells into a rat model of myocardial infarction and heart failure, we can improve its function. Next. And next. And as you can see, the second generation of cell therapy is what we call tissue engineering, in which we take our heart cell, mix it with scaffolding polymers, in this case, collagen, and we create what we call a tissue engineered tissue. In this case, we generate what we call like a calamari-like structure, beating structure, in which these are ring-shaped structures of human heart tissue that can actually contract. We use it also to measure force for drug studies and so on. And in the middle, you can see that our work with another biomedical engineering colleague at the Technion, Shulamit Levenberg, in which we collaborated and created not only a muscle, but also a vascularized muscle that will enable the muscle to survive after transplantation. One of the problems when using human embryonic stem cells, next slide, is that it doesn't come from the same patient, and therefore it can be rejected by our immune system. So many years after development of embryonic stem cells, a Japanese researcher by a name of Shinya Yamanaka, who won the Nobel Prize, taught us how to take adult cells from an adult individual and reprogram these cells like in a time tunnel back to becoming like a fertilized egg. So you can take a cell from a patient who is 90 years old and reprogram his cells 90 years plus 9 months back and generate the patient's own stem cells. And then we can use our technique, if you click, to generate the patient's own heart cells. As you can see in this diagram, you can see we got much better on creating heart cells. So now we can take a patient blood sample and end up with creating billion of the patient-specific heart cells, as you can see here. Next, just showing how in this project we are starting to close the loop. Our colleague who we collaborated on generating these calamari beating structures in the beginning 20 years ago, Professor Zimmermann from Göttingen in Germany, just took this approach this year, upscaled it to contain about 1 billion heart cells, made this patch, which is about 10 square centimeters, and implanted it on patients that have a very weak heart, who are candidates for heart transplant, waiting for a heart. So instead of transplanting the heart, he transplanted this patch, and initial evidence suggests that they may improve from this transplantation. So it took 20 years and many efforts from the idea in the bench in order to actually get it to the clinic after 20 years. Next. One of the problems with what I showed you is that we're getting heart cells, but as you know, the heart has different chambers. We have the upper chamber, the atria, the lower chamber, the ventricles, pacemaker cells, and so on. So the next stage was to learn how to push the stem cells not only become heart cells, but specific heart cell types. So for this, we collaborated with a group in Toronto in a collaborative program that was actually funded by Canadian Technion Society, and I would like to acknowledge very much the help. And this really made a breakthrough because now we can learn not only how to make heart cells, but how to make different heart cell types. So now we have all the Lego parts of the heart. For example, in this work, next slide, we were able to generate a pacemaker. And as you can see, if we put this pacemaker on a heart tissue, it can pace the heart. So the idea maybe in the future, instead of transplanting an electronic pacemaker, we can make the patient own pacemaker cells and replace the need for an electronic pacemaker by transplanting his own cells. Next. Next, Noam. And go next, go to the next. The next. Next. Okay. So before I end, the next thing is how we can use this for drug development. One of the problems is that when we develop drugs, they never are tested on human tissue until they reach a patient after investments of billions of dollars. And a lot of time after you invest $1 billion, your drug fails because they are never tested on a human tissue. What we suggest is now that we can make human tissue, in this case, human atrial cells, next, that can help us study what we call a patient in a dish. So as you can see here, we made like a small atria that can beat. Next, we can now test how electrical activity propagates in this tissue. So we can actually study like a small human heart in a dish. Next, and we can induce arrhythmias, as you can see here. This is, it doesn't project very well, but you're supposed to see something which is called a rotor. It's like a hurricane. This is what happens when you have fibrillation. So now we can finally study how to try to treat atrial fibrillation or ventricular fibrillation in a dish. We see how it is formed. We're pacing fast, and you can see how a rotor, how fibrillation is forming in this. So we have a way now to study and develop new drugs to treat the heart. Next. And the last slide, I want to see you another cool picture who shows how we collaborate in different fields. This is an idea comes from the neuro fields called optogenetics. It comes from algae. These are proteins that are ion channels that allow the algae to move away or toward light. What neuroscientists did, clone this protein, express it in the brain, and they can now manipulate the brain electrical activity by light. So we took it to the heart. Next slide. And what we formed is a new way to pace the heart by light. So you can see the heart beating. This is a rat heart. And now we are flashing the heart with a light. And each flat and each blue light flash paces the heart. So we generated a light-induced pacemaker, as you can see again. Next. And we can also do the opposite. We can also stop the electrical activity. So when we have fibrillation, this is a new approach for fibrillation. We can silence the electrical activity And here you can see pacing at a normal rate. This is a normal rate. Now we induce an arrhythmia. You see the heart is fibrillating, and instead of giving shock like you see in the movies, an electrical shock, we give light, and it stopped the arrhythmia, and the heart came to normal rhythm. So now, in order to now move into the clinic, we need engineers to allow us to generate light-emitting devices for transplantation. We'll need better delivery, how to deliver the genes that are responsible for generating these alter genes into the heart, and so on. So now that we have an unmet need idea and prototype, we can use all the capabilities of the center to try to move us forward to eventually affecting the patients. And that, I think, my final slide.
Thank you very much, Lior. So just to summarize what we heard, we've seen the 6 labs, AI and medicine lab. We've seen the bioelectronics, biochips lab that Amit talked about. Drug delivery and molecular therapy labs we haven't talked about, but we're free to ask questions. Same for the device innovation lab. Tissue engineering, which is what Lior was talking about. The healthcare operations lab, which is very interesting, but we haven't got to there. So thank you for your attention, and I will stop the share now.
Thank you, Noam and Danny and Amit and Lior for your presentations. We now have some time for Q&A, and some questions have already come through, and I think we'll start with those, but then we've been asked to move from location to location virtually. And one question which has arrived, which is about this evening, is about the question about length scale. And the question is, if you're already down, and Amit, I guess this is for you, down to 2 nanometer diameter DNA molecule, the question is if you have to take into account quantum effects when you make your measurements.
Okay, so thank you very much. Fortunately not, or maybe we don't. We are still, everything in biology and in physiology is at room temperature, and there are many electrons around. We have to understand the biostatistics and physics very well of soft matter, but quantum effects are not there, to my knowledge. No, but I'm happy to speak on everything else except that.
Okay, I understand. I do have another question here, and that's, I noticed examination facilities, and will the center treat patients? And I guess that's for you, Leo.
So
Or Danny.
So specifically in the center, the examination facilities are for research. For example, we have a program that we want to study about healthy aging in the center. Why some people, reach the age of 100 and are healthy and others not. So the idea is to follow many patients for many years and to take all the biomarkers possible. So this is one example where the patients will be in to draw blood, to take their physiological measurements and so on. Another is to test in phase 1 clinical trials and so on. But the idea is not to treat patients in the center, but for the projects involving human beings, have a place to perform these studies.
Very thorough, thank you. Kind of in parallel, but I think this is probably for Danny. The question is, who will be using the labs? So, and I guess it's a question about Rambam Technion collaboration. 1 + 1 is equal to 17.
So our vision that the lab will be a nexus for collaborations. So from the Technion side, students, researchers, anyone. On the Rambam side, we actually want the entire interprofessional representation from the entire interprofessional teams. So physicians at all stages, from initial training stages all the way to senior physicians, nurses, physiotherapists, nutritionists. We already have several projects that involve everyone, and they need to actually meet physically and interact. That's the way that we can actually progress. And as much as virtually meeting is nice, I think physically meeting and interacting, as Leo always says, I think Noam also, by the coffee machine while making coffee and waiting for the coffee to be made is the best way to move research forward.
So we're looking for the slowest coffee machines available.
Excellent. We've been asked to address if there are questions in each home, and I apologize, but I'm in Israel, so I'm guessing about whose home is who. And I might have gotten this wrong, but I was asked to first reach out to Edward. Edward, if there are questions from your group.
Yeah, is there any questions anybody has? Anybody want to ask any questions? I think you can move on to another house, Wayne, and then come back. If there are any other questions, I'll let you know.
But it's not.
Very well. Luis?
Yes, we have questions here at my house. Susan?
Yes, can you say who are the collaborators in the healthcare operations lab?
Can you repeat that? I couldn't quite hear it.
Maybe I'll take that one. The question was, who are the collaborators in the healthcare operation lab? So it's actually an interesting topic, kind of not really well thought about. When a patient goes to the hospital, there's a good reason it's called a patient, because he needs a lot of patience. So the matter of streamlining the visit to hospital and the experience is very important, and therefore there's a team in the faculty of what used to be called industrial management. Now they call them the decision science, data science and decisions. That there's a team that actually works on streamlining hospitals in terms of operating rooms, the ER, minimizing queues, making the whole process more efficient and more sufferable to the patient. I think anybody who's been to hospital, he knows that most of his time they are spent waiting, and we would like to minimize that.
And what about primary care? Are you interested in primary care?
Lior, would you like to say something?
Yeah, we're actually interested in all forms of medicine. What we envision is that the projects will come from the need. So if a primary care physician comes up with us with a clinical unmet need that sometimes it doesn't even have to know the solution. We'll try to form a group of interdisciplinary team who will try to do brainstorming and then come up with potential solution for this need. So the need could come from anything. It has to be an important need. We want all the needs in this center to come actually from the patient bed, come from a clinical question, not a theoretical biological question, but a question that can eventually help the patients either understand better disease or better treat diseases. So any type of medicine we encourage, as long as they come with important questions and that we can come up with a plan to try to answer them.
Any questions?
Thank you, Lior. I think we should.
Yeah. We have another question here.
Okay.
I have a political question. We see all of this, and we ask also the pamphlets that have been shared with us about all the innovations, and we wonder, can the world afford to sideline Israeli scientists? And yet we see that the world thinks it can afford to sideline Israeli scientists, and even not to work collectively in order to protect Israeli scientists who have gone the battlefield, and a lot of projects that have been wasted because their lives and their health has been affected. And this has been a terrible loss to the people of Israel and a ridiculous loss to the world. And so I want to ask, how is the Technion investing in PR campaigns and marketing efforts addressed to negative political attitudes towards Israel?
Yeah, so this one's a little bit off topic, and I will address it. I got this, and I got a variant of this question submitted as well before. And it's certainly something that I'm sure that everyone's sensitive about and worried about. And thank you for your concern. I appreciate it. It's not easy when you have, let's say, the Senate of the University of Waterloo trying to propose to divest from all research with the Technion, which happened about a month or so ago. It didn't go through the Senate, but it was proposed, and other places. And it's a little bit hard to take that type of approach from people that we know. Maybe people who don't know Israel, and I think the word knowledge is the key to understanding the solution. Is a little bit not easier to accept, but I think we have a better way to deal with that. And the best way to deal with it, like any form of bigotry, and this is anti-Semitism that's clear, and anti-Zionism, if you will, is education. And while we can't go out to the world and educate everybody, what we can do, and we are doing this, is to interact with as many people from all over the world who are not necessarily Jewish and don't know Israel and don't know the Technion. And we've had guests from a variety of places around the world who we've interacted with who at first didn't want to come here, didn't want to, were worried about it or concerned. And when they actually spent time with us, they were able to see the science, the amazing students, that's our secret weapon here, the amazing students, and to see what comes out of the Technion. And in a lot of ways, these people become some of our best ambassadors everywhere. So the Technion can't take the entire PR campaign of the state of Israel on its shoulders. It's really not our job, although sometimes when people suggest that we should, but we certainly can try to do our part, and we are. And we have a lot of efforts in educating people, reaching out to people, hosting people. A little bit less visitors at this time, but that'll change. It'll come back again. And I'm sure that many of you have heard about the First Year in English program, which is designed for people who are willing to come to the Technion and make the effort to learn Hebrew. I did it. It's not so hard. And to study as an undergraduate, I did it. And then can transfer into the regular program. And this program is not designed only for Jews. It's open for everybody. And we'll be delighted to have as much international participation as possible. And we're convinced that these graduates will be really important for us as ambassadors around the world. That's a small part that we're trying to do in PR for Israel via the Technion. Let's go back to the Wolf Center, if we may, and move to Neil, if there's a question in Neil's group.
Yes, hi, can you hear me?
Yes, we can.
Thanks, Wayne. Yes, we do have a question here from one of our guests. Go ahead.
I have been invited here today. I'm very happy to see what a country of 7 to 8 million people is doing for the humanity and for the mankind. And how can we communicate this message to what I do on an individual basis, mostly to the Muslim population surrounding Israel, so they recognize that they are missing something. I mean, say, what you guys are doing, it's beautiful. It seems to me the people who are surrounding you and which has gone now all the way to Iran. They don't realize it, they don't understand it. According to me, at the end, it leads to the mind business. How can we get this data out to that mind? The Jewish people are not working only for the Jewish people, they are working for the whole humanity.
Well, thank you for the question. Again, I'll field this, and I'll try to be short and to the point. It's a huge challenge, and again, not one that the Technion can handle only by itself in Israel. But if I may, I'd like to remind you that about 25% of our students speak Arabic. That's a value, that's the average number of students, undergraduate students at the Technion. 25, 1/4 of them speak Arabic. And they, believe me, they spread the message. And that has tremendous impact. We've also had courses, online courses, which are taught in Arabic and in Hebrew in parallel. And this has reached millions of viewers from our neighboring countries. So at least from a scientific point of view, this message does get out. And I'm sure this happens to my colleagues here, but I have to admit it hasn't happened to me this year. But normally I get applications from neighboring countries where it's not possible to come and get a visa into Israel, or it is, but they wouldn't be able to go home again. So the interest is real. By the way, that includes Iran. Requests to do a postdoc in my group from Iranian students. So I think that in some way we, at least in the scientific communities, we at the Technion are managing to do that.
Be a political question. It would be better to gain knowledge from them about the center if you have a question. We can follow the political questions later. We all
He's trying to listen to.
Yeah. I don't know if that was a question to us because I couldn't hear it.
No, it wasn't. It was a directive.
Thank you. Then I'll move on. Last but not least, to David. I was asked if there are questions from your group.
Do you have any question? Yeah.
Two short questions.
You want you have to go a little louder.
Louder.
Two short questions. One is aimed at Professor Gefstein. I was just wondering concerning atrial fibrillation. I understand that in today's world, one way of resolving it is using ablation. But I'm told that even though the ablation could be successful, the condition could still be there. Is it the case that with your development, the condition will be completely eliminated, or it's still going to be, existing? That's
so
one question.
That's a very good question, because it allows me again to brag about the Technion, because the ablation machine that people are using to treat atrial fibrillation was actually developed at the Technion. It was my PhD thesis with Professor Shlomo Ben Chaim. More than 25 or close to almost 30 years ago. And it's started at Technion, and now it's, for many years, it's the state-of-the-art technique to do ablations of arrhythmias in general, including atrial fibrillation. So it really transformed this field. So ablation actually, in the world, a lot of it started at the Technion. And so that's about bragging. But again, for atrial fibrillation, we are still far from being 100% in ablations. What we suggest here, and that's why I think there's a room for that, is that every time you go into atrial fibrillation, it will immediately stop, give you a flash of light and stop it. And the idea is to break the vicious cycle of atrial fibrillation, that even if your ablation doesn't work, it will stop AFib within a second after it starts, eventually preventing it from happening. That's what we hope, envision. So we think that there is a big need for treatments for atrial fibrillation. What we did with ablation was great, but from some type of patients, we're about 70–80% success rates. For other, we are less than 50% success rates. So we need better solutions, and we hope this could be one of it.
You had a second question?
Is it further commercialization?
I think this will be the last question, and then we'll wrap
Actually,
up.
there's one more after that.
Oh,
I promise
okay.
I would-
Two more questions then.
Commercialization of your, whatever you come up with, how does it work between Rambam and the Technion? Do you have a certain process, how to commercialize? Whatever
commercialization.
you innovate about, whatever product you come up with, and how, what's the process exactly? How does it work?
So both the Technion and Rambam have technology transferred units. And we have a very good understanding that any mutual project is shared by the Technion and Rambam. So a lot of times, good ideas are destroyed because fighting of intellectual property, and so on. But here it's all solved. The idea is that both institutions will benefit from it. So the idea is, after you have an idea, you can try to see if you can write a patent about it. There is a lot of ways in Israel to try to take early stage ideas and try to commercialize them. And we think we are in a unique situation to actually do this because we can come with the question. Because a lot of times I see ideas coming, let's say, from engineers from the Technion. They are great ideas, wonderful technologies, but they want to solve a problem that doesn't exist. The idea here is that we'll focus on problems and then try to find the best technologies to solve them. So that's, and then as Amit told you, try to streamline these ideas into products. And we'll try to support everything because we have from the beginning, the clinicians who know who will be the patient, people that are involved in regulation, clinical trials, and so on. So this effort can actually, I think, fast-track commercialization and has a huge opportunity for commercialization than the traditional ways. But that's my ideas. Maybe others will have other ways.
Well, I just add that since I was in charge of this for 5 years at the Technion, we have a very proven track history of strong collaboration between commercialization, hand in hand between the Technion and Rambam. And that includes everything that Lior said, but also including the concept that happens at every university where we have concepts which are technology ready, but they're not investor ready. And we know how to address that as well. And actually, the Technion is really known for that. So I think, we've managed to do this again and again, peacefully and directly and without conflict, which is not easy in the business. So I think it's very good. We had one more question that I don't want to leave because it was in the very beginning, and I sent it to Amit. And Amit, if you want to maybe rephrase it so that everyone understands it and then answer.
Yeah, sure. So it's a question from Ivan, and thank you for the question. Basically, the question is about DNA and protein. And in the devices, DNA and protein can be modified, and sometimes they have proteins attached to them, such as transcription factor. Are those lost or remain on the chip assay? And indeed, this is a very good question because one of the main advantages of looking at single molecules, like I showed you in this clip, is that we are sensing individual molecules. That means that we don't need to amplify the signal. Amplification, like you most probably heard about PCR amplification during COVID, we are losing a lot of the properties of the native DNA in our cell. We are actually sensing the native DNA from the cells. Endogenic molecules contains not just the bare DNA, but also proteins and other association. We sense them all. And in fact, this question came at a very timely coincidence for us. We just discovered a couple of weeks ago that we can sense native mitochondria DNA, which has not been sensed before because it has been, people has been using amplification, and we can actually see all the bound proteins. Now, mitochondrial DNA is associated with many different diseases, mitochondrial diseases, neurodegenerative diseases, and cancer. And this is something that, in fact, these kind of technologies really can open up a new window to sensing native DNA, native molecules in the cell. So this is a very good and timely question. Thank you.
Great. Thank you, Amit. I think Alyssa will stop now. I realize we've run over a little bit of time. I do want to, at this point, thank Noam, Amit, and Lior and Danny for your presentations, your drive and insight. This is really exciting. It's probably one of the more exciting projects, dynamic projects on campus. The melting pot of peoples coming together and approaching in different ways these challenges is really wonderful. And it's exciting. For all of you, I thank you for giving us your time to learn about the Wolf Center for Translational Medicine and Engineering. Like I said, I think it's really unique way for Rambam and the Technion to collaborate. And I think it's going to have a huge impact on science and a huge impact on healthcare and taking care of people. It's clear to us that none of this can happen without you. And again, many of you are already longtime partners with the Technion, and I thank you for everything you've done. For those of you who are just getting to know us, I invite you to partner with us and with the Rambam, of course, on this project in any way that seems to make sense to you. We are celebrating our 100 years this year. And yes, I'm saying we are going to celebrate it. I'm looking in a positive way that we will be done with this by the time of our BOGs. Rambam also has an event usually right a week before ours. And this year, we're celebrating 100 years of contribution to the state of Israel by the Technion. And I'd be delighted if all of you attend and join us here in Israel. And it would be a wonderful opportunity to meet these four gentlemen and their colleagues in person. If you have questions, you know how to reach Lynn or Alyssa or Sharon or Ali, Technion Canada, and we'll be happy to interact with you in the future. So thank you again, and I'll turn this back over to Technion Canada.
On behalf of everyone here at Technion Canada, we want to thank our Israeli partners. Thank you so much for taking time, especially during this very challenging time for you in Israel, for being with us here. I have to say that joining together as a community feels good and it feels important, even under what's going on. Our hearts are with you. Please stay safe, and we look forward to seeing you soon on the other side. Take care.